I'd like to start off by introducing Jake Beal. He's been doing our Interlab measurement project. And just so everybody understands, this was also an experiment for this year to try to find out if bacteria grows the same in China as it grows in Europe. Who knows? Jake knows.
Hi, folks. I have to say, this is actually the first time that I've been involved in iGEM, when Randy suckered me into organizing the new measurement track, and I have been blown away by what I've seen. And when we started putting together this Interlab study, we had no idea what kind of response we were going to get. So let me start, though, by saying what this is all about. Why we want to do an Interlab study at all is to get a baseline for how well things act the same all around the world. Measurement is, in general, a foundation for everything that we do in science, in industry, in society. Up there, I've got a picture of a really key early measurement device. This was the first clock that ever worked on a ship. Top secret 18th century military measurement technology. In synthetic biology, as all of you know, there's a huge challenge in understanding what the heck do our parts do? How much can we learn about what they'll do in our lab based on somebody else's lab? And also we wanted to know what equipment do people actually have that they can try to do good measurements with out there all around the world? And so we set up this interlaboratory study where we asked teams to build 3 constitutive GFP constructs. One was distributed in the package, 2 were a single pair of biobricks assembled. And if you weren't going to be able to do those because, say, you weren't working with E. coli or whatever, arrange an alternative with us. We wanted this to be very inclusive. We wanted to know what people could do. And basically just grow them up, measure their fluorescence, and then we had some extra credit for people to also measure cell-to-cell variation or compare green versus red and see if the ratios are similar, or run a bunch of other promoters too. So we set this out. We wondered how many teams will actually be interested enough in this terribly boring idea of calibrating our rulers to see if they're the same. We were blown away. 45 teams signed up from all around the world, including teams who didn't even have access to a system for measuring fluorescence. I was blown away when the Sumbawagan team from Indonesia wrote to us and said, the nearest fluorimeter to us is 1,000 kilometers away by boat or air. Can we participate? And we said, yes! They measured with cell phone cameras. Woo! Let's hear it for Sumbawagan. Of those 45 teams who signed up, in 18 different countries, 37 were actually able to return data sets to us. And when we put those data sets through a validity rubric and measured how consistent the results were, again, we were blown away. Across so many different laboratories, across so many different countries, we had a remarkable degree of consistency. And not only that, but we walked out of this with a baseline, a worldwide baseline, the first that anybody has ever been able to construct in the biggest interlab study that has ever been done in synthetic biology. What are the protocols? What are the equipments? When you say measure fluorescence, how differently do different laboratories interpret that notion? And so with this very rich amount of data that you out there in iGEM have gathered and contributed, where we're going next is, first of all, I guess we need to write a paper. We're working on this now. We'll see what happens when we send it in there. But this is some significant scientific data that you folks have gathered that's going to go out there into the literature. Next year, now that we know more about what people are capable of, what are the challenges in organizing something that's this big and complex with so many different laboratories with such different resources, we're going to put this together bigger and better with some more precise protocol instructions, simpler ways to do it, and we hope that we're going to get even more teams participating next year. Maybe we'll run the biggest Interlab study in the history of the world if most of iGEM signs up. So, I want to just close by saying a huge thank you to all of the teams who participated. Every one of the teams who returned data to us is getting an Interlab Study Award for having done so that will be at the awards table with everything else. So thank you to all of you who participated in Interlab.